I hope you all are doing well. So we are waiting for just our speaker facing some technical problems in his system. Uh, we will be on board within few minutes. Stay updated within the room live stream moderating system. So you can write your name along with serial number in the chat box for the attendance. Our team is taking the attendance. Hello, greetings dear Professor Dr. Ali Raza. Is my voice is audible to you?
Yes.
Okay. Dr. Ali Raza. Hello, dear Dr. Ali Raza.
Assalamu alaikum. Am I audible?
Yeah, you are audible. You are clearly audible. So, Miss Melina, you can start now. Thank you so much, everyone, for your patience. Melina, go ahead.
Thank you, dear
Okay, let me
Gulam.
confirm. I'll be sharing my presentation through my screen.
Professor, first I will do an introduction, then you can start. Is that okay?
All right. And what about the slide change?
Okay, so good day, everyone, and welcome back to the second day of the International Symposium on Biosafety and Laboratory Biosecurity Management. I hope you all enjoyed yesterday's insightful sessions. Today, we continue our journey of strengthening safety, securing science, and protecting lives with another lineup of incredible experts. As we dive into today's program, our focus remains on deepening our understanding of global biosafety standards and implementing effective risk governance in our laboratories. Thank you all again for joining us today. So, to kick off our sessions today, it is my distinct privilege to introduce our next distinguished guest speaker, joining us from Pakistan, Dr. Ali Raza. So, Dr. Ali Raza is a highly accomplished molecular biologist and a certified biorisk management professional. He currently serves as an assistant professor of basic research at the prestigious Liaquat University of Medical and Health Sciences in Pakistan. So, with his deep expertise in molecular biology and his specialized certification in virus management, Dr. Ali Raza is at the forefront of driving safe, secure, and cutting edge laboratory practices. Today, he will be sharing his invaluable insights and expertise with us. Please join me in giving a warm virtual welcome to Dr. Ali Raza. Dr. Raza, the floor is yours.
Thank you so much for giving me the chance to speak. First of all, I'd like to thank VitaNova for organizing such an informative symposium on biosafety laboratory biosecurity management. Today, I'll be talking about biosafety. The topic of my presentation is biosafety rules exist. Do we follow them? So, biosafety rules, guidelines, policies were already made, were already in practice. But after COVID pandemic, it becomes the highlight. It becomes a matter of concern that biosafety rules, policies exist, but do we actually follow them? Because there are so many breaches, so many lapses in the biosecurity applications. Like there were policies, but implementation was not that much. So yes, starting with our presentation. First of all, before starting biosecurity, I'd like to clear some definitions and some terms. What is risk? Basically, it's an undesirable event that involves a specific hazard. And in response to that hazard or threat, there is the consequences. Now, consequences may be low, consequences may be high. It depends on the likelihood of a hazard. So for that purpose, you should know what hazard is. If you don't know about your hazard, it's very much difficult to know what is the likelihood of that hazard or what is the probability of the undesirable event. So you must know your hazard. Then you must know what is the probability of that or what is the likelihood of an undesirable event. If you come to know about things, then ultimately you'll be knowing your risk, that what is from that hazard and what will be the consequences. Let me give you a general example, general life example. If a person is crossing a road and a bus is coming with a speed of 80 kilometers per hour. So at this time, hazard is on bus. And what is the probability of this event, undesirable event? Because the bus is moving at a speed of 80 kilometers, and if it hits you, the consequence is going to be the worst. In the place of bus, if there's a bicycle, of a person riding a bicycle and you are crossing a road, so obviously the consequences will be low, because the hazard that is coming, that is different as compared to the bus. Similarly, in our labs, when we are working with the pathogens, there are different type of pathogens we are dealing with our labs in our daily life. So every pathogen has its own likelihood, on risk, and it has own consequences. Not only pathogens, but hazard can be a person also. Laboratory staff, untrained staff work in our lab. Or suppose you are a well-trained staff and with you a new trainee is working. So he or she may be a hazard also. And there are glassware hazards, electrical hazards. There are so many hazards. So hazard can be anything. So you first know how to identify your hazards. Now, for that purpose, we do risk assessment. Yes, please, moving to the next slide. Assessment. And what is risk assessment? In simple terms, risk assessment is basically the process. It is a process that is used to establish the controls in order to prevent harm from hazard. If you are starting any process, suppose for example, COVID-19. We have the sample of COVID-19 virus. We have a nasopharyngeal swab, and you are dealing it with first time with the nasopharyngeal swab. So you will do risk assessment. What you will do, we will be discussing these steps in next slide. But before starting any new procedure, you will do risk assessment. When there is staff in your lab coming to you and working with you, then you will do risk assessment. When you are working in at a place, you are doing a procedure, but your building has been shifted, you are going to a new facility, then you have to do risk assessment again. Like similarly, same example goes like I gave you in that a person crossing the road, he analyzes, he assesses that if he crosses the road, will the bus hit him or he will cross the road. So he is doing assessment. Or whenever we do any procedure in lab, because every lab has its own risk. Risk is not same for every lab. It varies lab to lab, it varies institution to institution, it varies country to country. So risk assessment is never same. Suppose I have done risk assessment of a particular procedure in my lab, and same procedure cannot be applied, same thing cannot be applied to other labs, because my lab is have sufficient resources, sufficient personal protective equipment. My lab is facility design is well established. I have proper BSL2, BSL3 lab. I have sufficient students. So my risk assessment will be different. If a lab that is deficient in resources, that is deficient in technologies, the risk assessment will be different for that lab. So moving to the please change the slide. First. Yeah. So this is the cycle or a process. It has been adopted from CDC Biological Risk Assessment course. How we identify or assess the risk. Number one is we have to identify the risk and hazards. That what, where, and how is the work occurring, who is involved in the work, and what can go wrong. So number one thing is you have to identify the hazards and your risk. Then after identification of hazards, you have to evaluate the risk. That either the risk is acceptable or unacceptable. If risk is acceptable, we proceed with our procedure. If risk is not acceptable, then we determine controls to reduce the risk to the acceptable level, because risk is never zero. So we determine the controls, and there is a specific hierarchy of controls. Number one thing is that elimination. that you eliminate the risk hazard. That is not always possible. So number thing is substitution. We go for substitution. We substitute or replace the hazard. Suppose we are dealing with glass in our labs. So our hazard is glasswares. We can substitute it with the plasticware equipments as much as possible, because now we have we are substituting by this. But control is our engineering control. Yes, engineering control is like you isolate the people from the hazard. There are several means by which we can isolate the people from hazard, like by using biosafety cabinet. We deal all the pathogens, we conduct all our procedures inside the biosafety cabinet. So we are isolating the pathogen from the people. Or as per the risk assessment, we conduct our experiments in BSL-2 lab or BSL-3 lab. There should be a negative pressure inside the lab, like more air should go inside the lab rather than going out of the lab. That is called the negative pressure. Then the fourth one is the administrative control. Administrative control means implementing policies, standard operating procedures inside our labs. And last one is PPEs, personal protective equipment. Personal protective equipment includes our mask, goggles, face shield, or Tyvek suit, shoe covers. These all are the protective equipment that are used based on risk assessment, because not every PPE is worn in every procedure. Sometimes we have to reduce PPE, sometimes we have to enhance the PPEs. So it is not necessary that suppose we are working in a closed system, there is an automation in our lab. We are performing hepatitis B, hepatitis C, or a PCR on an automation, an automated analyzer. So obviously our risk assessment, based on our risk assessment, PPE's usage will be reduced, will be low. But if you're doing manually in a safety cabinet, or like we are extracting DNA manually and then we are making master mix, then our PPEs will be different. We are doing powered PCR, our PPE selection will be different. But if you are doing TB culture, our PPE selection will be different. So it depends on our risk assessment. So not every time N95 mask can be worn. Not every time you cannot wear a Tyvek suit. Sometimes you can just wear a simple gown and a surgical mask. So again, I would like to say that it depends. It depends on our risk assessment. Please, next slide.
Yeah, here is his next slide. What is
Yes, sir.
biosafety and
So
biosecurity?
now coming to our main topic. Yeah, we are coming to our main topic, biosafety. What is biosafety? So in simple terms, it is used to prevent the unintentional exposure to pathogens and toxins to their accidental release. According to WHO, this biosafety depends on the containment principles, technologies, practices that are implemented to prevent unintentional. Like in yesterday, Professor Usman gave a wonderful presentation on lab management system and biosecurity. In biosecurity, he mentioned intentional exposure. So in biosafety, it's unintentional, because you never know how you're going to get infected from the pathogen, or your environment going to get contaminated, or the surrounding people going to get contaminated. So biosafety is all about prevention from an unintentional exposure, and this is it comprises of three practices: primary barriers and secondary barriers. We'll be discussing it in next slide. So, like I said, it comprises of three work practices: primary barriers and secondary barriers. And their combined purpose is to protect laboratory personnel, community, and the environment from the infectious agents. Now, what are biosafety work practices? It's all about our good laboratory practice. It's all depend on our good laboratory practices, and it includes like no eating in lab, proper hand hygiene must be followed. they're following the SOPs, decontaminating lab properly before and after. Like when you start your work, you start your procedure, you have to clean your surroundings, to clean your workbenches. And when you end up your work, end up your procedures, you again clean up your surfaces, your workstations, everything. It's not like that you tomorrow or afterwards, because by doing this, you are contaminating your lab. You are exposing others also. So please, next slide, please. Actually, so primary barriers include our biosafety cabinet, our safety equipments, and protective equipments. Biosafety cabinet provide immediate protection from pathogen, and it's a barrier between the pathogen and our lab personnel. Safety equipments like centrifuges cups, like when we are using centrifuges, the centrifuges must have lids, so that while centrifugation, the circulated due to the centrifugation force, aerosols generation occurs. So aerosol should not circulate in our surroundings, in your environment. So you should have a centrifuge cups also, sealed rotors. Like I told you, personal protective equipment includes gown, laboratory coats, gloves, face shield, eye protection. So these all are the protective equipments, but we decide based on our risk assessment that which PPE should be worn and which should not be. So moving to the secondary barriers. So secondary barriers basically provide protection for the environment outside the laboratory and the surrounding community. Primary barrier was actually protecting the immediate protection between the personnel, lab personnel and pathogen, while secondary barrier provide immediate protection between the surrounding outside the laboratory, like our community. So it includes facility design. Facility design means controlled access zones, dedicated wash areas, and controlled access zone means while your procedures is being carried out inside the lab, so that lab must be at the door of that lab must be closed. Only the authorized persons can enter the lab, and no unnecessary movement should be there. Like people come and go with unnecessarily moving inside and outside the lab. By doing this, so they are contaminating the environment. By opening frequently doors of the lab, they are contaminating the air also. So the facility design, then ventilation. There should be a specialized air handling system, especially the negative pressure. Like I said, more air should flow inside the lab. Doors should open inside rather than flowing air going outside the lab. That is called the negative pressure. And negative pressure is maintained to prevent the contaminated air to go outside. If it goes outside, it will be contaminating the environment. Then structural features are also there, and then waste facility, like whatever the waste is being generated inside the lab must be decontaminated inside the lab, must be autoclaved inside the lab, and then it should be sent for incineration. Like you cannot directly send waste for incineration without decontaminating it or without autoclaving it. That is also one of the protocol, that you are preventing the environment, you are preventing a community also. Next slide, please. I guess my slide is visible. I have uploaded just.
Yes, it is all visible.
Yeah, that's good. So I guess I must. I would like to request host to please load my slides so I can easily change the slides. Apologies for the interruption. Yeah, that's good. Thank you. So we are talking about now, when we talk about biosafety, there are 4 biosafety levels: biosafety level 1, biosafety level 2, biosafety level 3, and biosafety level 4. Now, how to decide this biosafety level, like which biosafety level is suitable for which kind of pathogens? So WHO has classified 4 risk groups: risk group 1, 2, 3, and 4. So risk group 1 are those pathogens. So different pathogens are included in these groups. The pathogens that are included in risk group 1 are those that are like individuals and community have low risk. Means they are not that much harmful. They are not more prone to cause harm, or they don't cause any sort of animal diseases. These are in risk groups 1 pathogens, and they are dealt in biosafety level 1. Biosafety level 1 is the basic laboratory. Then comes risk group 2 pathogens. The pathogens that are included in risk group 2, they pose moderate risk to the individual and low risk to the community. So they can cause disease, they can spread, but there is an effective treatment present for risk group 2 pathogens, and they are dealt in biosafety level 2 lab. The pathogens that are included in risk group 3 pose high risk to the individual, but low risk to the community. They can cause the serious disease. They can cause the serious disease, but treatment is usually available for these kind of pathogens, like Mycobacterium tuberculosis. It causes serious diseases in individual, but there is a treatment available, so obviously community is at low risk. But these kind of pathogens are dealt in BSL level 3 lab. And risk of 4 include deadly fatal pathogens that cause fatal condition, that can be fatal for the individual and for the community also, and they are dealt in BSL level 4. And BSL level 4 is the maximum containment facility or maximum containment level of biosafety, which has a separate building, which is built at a separate place, separate building. There's highly restricted site. Not every person is authorized to enter in these kind of labs. They have a proper authorization, proper training for that. So these risk groups are related with biosafety. Now comes biosecurity. Like I said, biosafety is unintentional exposure. We have to prevent unintentional exposure, but biosecurity is to prevent intentional release of pathogens. Like, in simple terms, if I say biosecurity means we have to protect ourselves and community from pathogens, but biosecurity means pathogens must be protected from humans, because human can misuse the pathogens that can cause serious threat to the community. Like we after COVID or in COVID, during COVID pandemic, we heard it's a bio war, bioterrorism. So what is bioterrorism? It's due to biosafety and biosecurity breaches. There is a lapse, there is a breach in biosecurity or biosafety, obviously a bioterrorism can occur. So in biosecurity, we prevent pathogens from humans, and it has 5 components. It has like physical security, personal security, material control and accountability, transport security, and information security. We'll be discussing it one by one in our next slides. But most important is that why we need a biosecurity program. When there is a biosafety in place, then why biosecurity? Because we have to deter criminal activities. We have to overcome our bioterrorists. So like from acquiring pathogens, or like we have to protect pathogens from these bioterrorists and criminal persons, so that they do not pose any sort of harm, or they cannot misuse that kind of pathogen for or they should not use those pathogens against humans, agriculture assets, or against animals, anything, because ultimately it is going to affect our economical situation also. Suppose if any pathogen that is harmful for the environment is stolen from any facility and that is being used against our agricultural resources, any virus or any bacteria, so what is going to happen? Our agriculture down, and ultimately it will be affecting our economic situation also. So, and it also, biosecurity program is also required to protect the employees and co-workers. It also required to improve the security of our laboratory and the assets of laboratory. Here, I would like to give you two major examples. There are several examples, several historical examples of lab incidents, but there are two common examples. Like one in 2014, CDC mistakenly shipped a sample of highly pathogenic H5N1 influenza to a BSL-2 lab, where it was used in experiment until researchers realized the error. And second was in 2015, where anthrax virus, 194 live anthrax virus was distributed to labs where they were not supposed to be. But fortunately, there were no incident reported, no any sort of harm reported. But that was the lapse between because of biosecurity and biosafety. So if I say that biosafety and biosecurity are synergistic, so I may not be wrong, because these both components work together. Our web practices are good, or primary barriers are sufficient, secondary barriers are also there, but our personal security is missing. Our physical security is missing. So what is going to happen? That pathogen or that virus or that bacteria is going to be misused. It is going to be in wrong hands then. So biosafety, biosecurity, both are synergistic. One cannot exist without another. So biosecurity, like I told you, there are 5 biosecurity components. I will go through the components. One is physical security. Physical security simply means like you have to install a CCTV camera. There should be a protection according to the risk assessment. That what kind of virus? It's not necessary that you are working on hepatitis B and C and you are getting Z+ security in your area. Risk assessment is done to avoid the misuse of the resources, safety or biosecurity. So based on our risk assessment, we choose what kind of security we require. So if it's high containment facility, so there will be a great protection, CCTV camera, alarms, auto-lockable doors. So there should be, and visitor access, like there should be a proper escort visitor entering inside the lab. At what time he's entering, there should be a proper record of that, and what time he's leaving, and what activities is being conducted by that person inside the lab. Suppose different students are visiting different labs. So there's a logbook where it is all maintained and they are monitored through the CCTV cameras or the different logbooks. Second thing is personal security. It is vital that individuals working with or access with pathogens are responsible, reliable, well-trained, and trustworthy. And this is very much important because the personal security matters. You never know the person who is working on a pathogen having good intentions or wrong intentions. He or she may misuse that facility or misuse that pathogen, misuse that virus also. So for that purpose, personal security, background checks in high containment facility, background checks are done by security agencies. ID cards are displayed, are given, special ID cards are given to the personnel who are working on specific pathogens. Their training pattern is evaluated. Are they capable of dealing with the specific pathogen or not? Suppose a person who don't have any experience and we send him to the lab where TB culture is being performed. So what will happen? Ultimately, this will be bringing a disaster to us. Then comes the material control and accountability. Again, it is very much important that the material that has been used in our lab must be documented, whether it's for pathogens, whether chemicals, whether reagents, whatever the resources of labs are. So Even the pathogen inventory is also maintained, that how much pathogen, how much ml strains we have, who is working on it, consumed, how much is left. So everything is logged inside a book, or whether it's a compressed system or there's a logbook. So everything is documented, and there's proper chain of custody also. So all pathogens should have an associated accountable person, because when multiple persons are working, it's very much difficult to be accountable, to make anyone accountable, or ask anything, any mishap incident. So if a single person is accountable for that, he or she will be responsible for all the mishaps, all the threats. So because he will be distributing the pathogen or strains among the lab workers, and he will be taking the record of that also. Then comes transport security. Yes, we kept all the measures. We followed every protocol. We are following each and everything. We are having a good trained staff. We are having good physical security. We were having material control and accountability. But suppose we have to transport the sample to another facility. Sometimes we transport sample within institutions, or sometimes we transport samples to other facilities. Like suppose we want to sequence anything, but we don't have sequencing facilities. So after extracting DNA, RNA, we have to send samples to other facility for sequencing. So there should be proper transportation system, proper packaging. Number one thing is packaging of sample. If it is highly contagious sample, so it must be sent in triple packaging. Triple packaging means number one is primary tube in which the sample is present. Second one container is we put that primary tube in the secondary container, and we keep cotton or absorbent material around it in the secondary container, so that if there is any sort of leakage, that must be absorbed inside that secondary container. And then that secondary container is being transferred to the tertiary container, and that is called triple packaging. Then before and after transporting the sample, there should be a proper record of that who is shipping the sample, where we have to ship the sample, at what time we sent, what time the sample received, who was the receiving person, was there any sort of leakage during the transportation or not, were SOPs followed or not, temperature was maintained or not, and there was a specific chain of custody or not for that sample. So by doing this, we can protect many things. We can protect many. And last one in biosecurity comes information security. Information security is to establish the policies for handling sensitive biosecurity information. Suppose, again, I will like to give example of high containment or high maximum containment facilities, that we do not share facility plan of that facility. Then access codes are not being shared with anyone, because it's the information. If it's become public, it's going to be a disaster for us. Then agent inventory and storage location are also kept secret. Personal information must also be kept secret. Test results and research finding. Research finding are shared need to know basis, and not everything is shared. Some information is shared, not everything. So we have to protect information from unauthorized release and ensure that the appropriate level of confidentiality is maintained. So these are different information security, like different labs have different type of information criteria for storing information. Some keep in PC, some keep in hard copies in terms of the files or like handouts. So there are different modes of storages. We talked about biosafety and biosecurity. Above these things, many breaches, many protocol violation happens. Why is that? We have standard operating procedures for everything. We have documented SOPs are the documented processes for every procedure. Like we are performing PCR, there will be an SOP for that. We want to extract DNA, there will be a specific procedure for that. we want to decontaminate the waste, there will be an SOP in established lab, well-established lab. There are SOPs, but where thing goes wrong is of behavior. That's why standard operating behavior is also mentioned in my slide. Behavior of a lab person matters. We have the documented SOPs. It is kept in our shelves, but I have read all the SOPs, the procedure I am doing, the procedure or the pathogen I am working with. I know the SOP for that, I read everything, but I don't want to follow. That's my behavior. So my behavior is not allowing me this. So there will be a breach in our biosafety and biosecurity. So SOP is there, plan is there, but behavior should also be there. So plan plus behavior is equal to outcome. Not only plan always works until unless you have any sort of behavior of implementing that plan in your procedure or your application. So standard operating behavior matters. These all breaches in biosecurity are due to our behaviors that we don't want to adopt. And due to that behavior, laboratory-acquired infection occurs. And over 80% of laboratory-related illnesses are unable to be tracked for a specific source. Person who is working in laboratory, a person working in any sort of virus, practices, we have to follow good laboratory practices. But SOP was there, but a lab personnel working on a pathogen, and suddenly a phone call rings, and he picks up the call with the same hand, with the same glove that is contaminated, and he is using that phone to pick up the call and then that phone back into his pocket. So, what is the use of SOP? Unintentionally, he has contaminated himself, and obviously he will go home. He will contaminate the community and environment also, because his cell phone is contaminated now. So good laboratory practice matters in it. You have to follow the SOP. You should have a behavior to implement to adopt the SOP. And due to if we talk about procedural and behavioral errors, sharp injuries occurs. And why sharp injuries occur? Because recapping is not recommended, but people are doing people are still recapping the needles. So due to the recapping of needles, like sharp injury can occur. Majority of lab cases are of sharp injury. But why they are? Because we are not reporting it. Incident reporting is very much important. So whenever the lab incident occurs, you should immediately report that incident so that corrective actions may be carried out for that purpose. Then improper removing of PPEs. I will discuss in end of my slide that how to remove the PPE. We call it doffing. We call it doffing. So improper for PPE removal, and then mouth pipetting. Obviously, mouth pipetting is recommended, but in some of the labs, people do mouth pipetting. They, in simple terms, if I talk in layman, people say nothing would happen. Nothing is going to happen. It means their behavior is unconscious. They are unconscious about the outcome or the consequences, that what is going to happen in if they're going to mouth pipette anything, or they're doffing off their PPEs improperly, or any sort of. So you have to immediately clean the slash. You are waiting to procedure to end up, and then you will say, some people, I have observed these things practically, that people say, okay, at the end of procedure, we will clean up the spill. No. As soon as the spill occurs or splashes is spread, you have to clean it immediately. So by doing this, you can keep the environment safe. Then aerosol contamination failures. As you can see in this picture, unintentional aerosol generation. There's a centrifuge, there's a tube inside it, but no lid present over it. So when centrifuge will start rotating, it will be generating aerosols or any sort. Suppose if a tube breaks inside the centrifuge, again, it may because it is a risk and the consequences disruption in network. Am I audible?
Yes, yes, you are now.
Am I audible?
Yes.
Ms. Melina, am I audible?
Yes, yes, you are. Can you hear me? Can you hear me? You can continue, please. Can you hear me?
Mr. Murtuza and
I'm sorry to the audience. We have some network issues here. We need few moments more so our Dr. Ali Raza will come back. I hope so. You can ask some questions while that time. So we will answer them at the end, I hope.
Miss Melina, Mr. Mansoor, am I audible?
Yes, you are. Can you hear me now?
Yeah, yes. Apologies, there was a
Yes,
disruption in
yes.
net.
Yeah, network issues. Please
Uh,
continue.
yeah, obviously, I'm almost about to end my presentation. Yeah. So I was talking about some waste transfer. There may be many problems like inadequate waste inactivation. Like I said, the waste that is being generated inside the lab must be decontaminated, autoclaved inside the lab, and then it must be properly segregated. What I mean from segregation is this: contaminated waste and non-contaminated waste must be separate. Glassware must be separate. Contaminated glass, everything must be separate it uh, because it will avoid the wastage of resources, unnecessary wastage, unnecessary finance burden. So we have to properly segregate office waste paper and all the things that are non-contaminated must be packed in separate box, separate container. Glassware must be separated in a leak-proof container. Contaminated waste must be separated in a separate container. So all things must be separated. Then comes our engineering facility faults. These are also very much important because when our lab design or lab facility is not proper, suppose there's no proper ventilation, no negative pressure inside the lab, ultimately the contaminated air will go more out of the lab and it will be contaminating the environment around our laboratory. So there should be no ventilation breakdown. There sometimes degraded infrastructure also contribute in biosafety and biosecurity breaches or in contaminating our environment. Like there is cracked drainage pipes, there are faulty air filters in our lab that must be immediately corrected or repaired. Or sometimes, like I said, in high containment lab, there are auto-lock or interlocks, auto-lockable doors. So that also lead to our biosafety and biosecurity breaches. Then comes administrative and oversight issues also. And these administrative and oversight issues like pathogen misclassification. Suppose we have to do, there is COVID. I would like to give example of COVID. We want to do PCR culture. Sorry, we want to do COVID culture, and we are performing it in BSL2 lab. Although COVID culture must be done in BSL3 or at least BSL2 plus with some additional advanced PPEs. So rather than doing COVID culture in BSL3, we are performing it in BSL2 because it was misclassified. It was labeled as risk group 2 pathogen. So it's a misclassification basically. And then poor training. A person is not trained on PCR, but he is working on COVID culture. He is performing COVID culture and he don't have proper or adequate training. So again, it's an administrative oversight issue. I will give you now some example. We are coming about the conclusion of presentation. Like suppose, what's wrong with this picture? There's a lady working in a lab in a biosafety cabinet, but like I said, there should be no mess inside the biosafety cabinet, but hands-frees are present on the grill because these grills are very much important. Air that is being filtered by the HEPA filter inside the biosafety cabinet recirculate through these grills, but their whole grill is blocked. Then wearing jewelries inside the lab, there's cold drink also inside biosafety cabinet. Lab coat is tied around the waist. So these are male practices. Already, obviously, SOPs are there, but our behavior is not adopting it to implement. See, this is how we work in biosafety cabinet, clean to dirty. This is the specific protocol. We always work from clean to dirtier side. don't directly clean the dirtier portion. We first start from the outer side or the cleaner side, then comes towards the dirty side. So we always work from clean to dirty. As you can see in this picture on left side, these are chemicals that are clean. Center portion is our working area, and the right one is the contaminated material that are being waste. That means there's a waste bag over there. If you look at this picture, this is also a common practice I have observed. That are the practical experiences that I'm sharing with you. That a person working in a lab or hospital coming out of this wearing a shoe cover. Sorry. So shoe cover is always removed whenever you are leaving the lab. It must be removed. So person wearing a shoe cover, again, what he is doing, those contaminated shoe cover is in market. He is moving in market, contaminating everything because of these shoe covers. Gloves and lab coats, these belongs to our labs. These belongs to our hospitals, not in for the market. So lab coats and gloves must be doffed, taken off whenever you leave your lab. It must not be worn inside the market in public areas. Again, they are contaminating the environment. These are the minor things that we ignore and we contaminate our environment. Now, as you can see in this picture, A and B, females who are wearing scarves, so their scarf must be inside their lab coat. As you can see the picture A, the lab coat scarf is inside her lab coat, but if you see picture B, the girl with the B red mark, her scarf is over the lab coat. Obviously, this will be creating disruption or disturbance inside the laboratory. Her scarf will be contaminated. The glasswares or the equipments or the consumables or the pathogens that or the samples that are on our working benches that may also come in contact with the scarf. So our scarf must be inside our lab coats. This is an example of centrifuge. There should be no open caps. All caps must be closed, and there should be a lid over the centrifuge while you are centrifuging the sample. So this is the donning and doffing procedure. While we are donning means to wear PP. Everything is clean, everything is new, you can wear, but there's a sequence that you can wear anything. That's not the problem. But while doffing, while removing the PP, it matters. You remove the most contaminated thing first, and that is your gloves. Gloves are the most contaminated part of our PPE that is removed first, then comes goggles, then gown, then face mask, and if you are removing shoe cover, then shoe cover, and then wash hands. With if they are alcohol-based sanitizer, sanitize it, but better is soap and water. And this is a specific biorisk management study. I will already discuss. I will quickly go through that. First of all, you describe your work activities, identify the hazard, and determine the risk. Once you determine the risk, you decide whether the risk is acceptable or not. If risk is acceptable, then you will proceed with your work. If not, then you prepare a risk control action plan, implement, and start your project. So in last, it's a good saying. Then it's taken from Alice the Wonderland. A cat says, "Where are you going?" And Alice says, "Which way should I go?" The cat says, "That depends on where you are going." Alice replies, "It does not matter where I go." The cat replies, "Then it doesn't matter which way you go. So it depends on you. You have the SOP, you have the protocols. Either follow it or not. But remember, you are going to contaminate whole environment, even your families, if you are disobeying or not following the SOPs. These are the references for my presentation and acknowledgement. I'd like to acknowledge our Honorable Professor, Vice Chancellor, Professor Kamdinojan, who is the Vice Chancellor of Liaquat University of Medical and Health Sciences, for promoting me in my educational journey, because he is very much supportive throughout for everyone who is willing to do good in healthcare setup. So he always promote education. Then Professor Saeed Khan, he is one of my mentors. He is the program director of Provincial Public Health Lab Sindh in Pakistan, and he is also head of molecular pathology. I have learned many things from Dr. Saeed Khan Sahab in terms of biosafety, also in administrative point of view, also many things. He has guided me at every step And then last, Mr. Shehbaz Ahmed, he is pathologist in common management of AIDS, TB, and malaria program, Ministry of National Health Services and Regulation. He is also one of my mentors, and he has guided me in many things according to biosafety and biosecurity. Thank you. Any questions?
Yes. So, Dr. Ali Raza, thank you so much in first place for this wonderful and highly informative presentation. Your insights into molecular biology and biorisk management are incredibly valuable, and you've given us a lot of fantastic points to think about. We truly appreciate you taking the time to share your expertise with us today, and it was an absolute honor to listen to you.
Thank you so much.
We
I'm
have
highly
a few. Yeah, you're welcome. We have few questions. So, can I read it to you?
Yeah, sure.
Okay, so first question is, can this relate to the Chernobyl incident?
Sorry, I cannot hear you. Can you repeat the question?
Yes. Can this relate to the Chernobyl incident? Can you hear me?
Miss Medina, I actually can't hear you. No, I'm sorry. There's some sort of distortion. Can you write it or type, or can you repeat it, please? Because I cannot hear you properly.
Can you hear me now?
Yes.
Okay, so the question is, can this relate to the Chernobyl incident?
Obviously, because see, like I gave you minor examples. I gave you minor examples of biosafety and biosecurity breaches. A single mistake can lead to a bigger disaster. A single mistake.
Yeah.
Reporting, we can rule out the root cause, and we can implement safety control measures, and we can avoid such kind of act in future also.
Okay. Can you hear me still? Can you hear me?
But yes.
We have second question. How to incinerate samples
Yes.
within the lab?
Basically, there should be a proper incineration facility, and before incinerating sample, make sure, because sample are many. Sample can be in liquid form. Sample can be a tissue form also. Sample, like there should be a liquid sample like blood samples. Okay, there are some chemicals also with the chemical sample mixed inside it. So it depends what kind of sample you are talking about. So first of all, waste segregation is important. Segregate waste according to the defined protocol, and then incinerate it. Not directly incineration is recommended.
Okay, thank you. Third question is, someone write that our labs are facing challenges related to limited funding and inadequate containment facilities. Sometimes we find it difficult to adopt some of the strategies mentioned. Please, we need your advice.
Challenges obviously depend on your workload. There is a BSL2 lab, and a lab itself knows its capability, that what kind of and pathogen the lab can deal with, and what are the financial resources of that lab. what are the administrative framework, what is the administrative framework of that lab. So they should not go beyond that. Likewise, in COVID-19 pandemic, many labs were there who were not capable of performing laboratory tests of COVID-19, but due to the government policies, financial resources, manpower was provided to those labs. So labs should do the risk assessment before going or before taking any. They should analyze their administrative framework and then go for that.
Oh, thank you. Next question is
And
how to.
it includes the trained staff also, not on the financial resources. Yes.
Yeah.
Yes,
Yeah,
please carry on.
yeah. Sorry. So how to implement biosafety in labs already constructed?
For implementing biosafety, first of all, proper adequate training is required, proper certifications. Like I've been trained by multiple sources. First of all, I am a virus management professional from International Federation of Biosafety Association, 𝗓𝗏𝗏, Canada. Then I have attended different trainings, master training programs also at national level. Like there's an NGO, 𝗕𝗏𝗏. I have been trained by that NGO also. So first of all, adequate training is necessary. Then you have to make a biosafety committee inside your institute. That's called institutional biosafety committee. And in that biosafety committee, there are multiple people with multiple subject experts. And among them, there's a biosafety officer also. So they combinedly make up the policies, they check, they approve, and they circulate the policy among the institute, depending on the nature of work they are conducting. Because not every policy is for every institute. So every institute has its own risk assessment and its own policy, but following the international guidelines and standards and the national guidelines also.
Yeah, exactly. Thank you. Actually, next question is maybe related. So HIV is one of the most risky samples. Please compare it with our immunity as how much our CD4 cells are enough to survive with this disease. Once I heard that if you're 1,200 plus, you're safe for whole life.
It depends on type of pathogen that you are talking with, and second thing is your vaccination. That are you vaccinated or not? And in vaccination, many people do that. There's different doses. Some doses are quarterly, some are yearly, and there comes a booster dose. And booster dose doesn't mean that you are 100% safe. So after booster dose, there is specific time. I exactly don't know, so I will not say anything about it. After booster dose, again, some doses are required after several years. So it varies person to person, and it depends. It's not for same for everyone.
Yeah, definitely. I thought
So
that
vaccination
also.
is
Thank
important.
you. Yeah, vaccination is important, really. Thank you. And actually, last question here is if accidentally get
Yes.
infected by an unknown pathogen, what precautions must we follow
Three.
at that time?
Please repeat the question, please.
Yes, of course. If accidentally get infected by an unknown pathogen, what precautions must we follow at that time?
Uh, First of all, pathogen is never unknown. When you are working with any pathogen, like I said in my presentation, you have to gather the information first. That what kind of pathogen you are working with, what are the procedure to be conducted, and we have to see for the possible exposure and consequences of that pathogen and the treatment plan available for that. Let's say you get exposed by a pathogen, immediately report it to your supervisor, the work your lab you are working inside. You must report it, the incident, to the supervisor, and the supervisor will call for the immediate medical assistance. that is required for a specific pathogen. So incident reporting is mandatory.
Yes, definitely. Also, people are asking if they can have this wonderful presentation, if you can share with them.
Uh, yeah, sure. I will be sharing. I will give it simply. They can reach out to me also, and if you say, I will share it to you also. You can. It's for the people.
Oh, great. Thank you very much. So that's it with the questions. Dr. Ali Raza, thank you so much again for this wonderful and highly informative presentation. I think all of us taught something new today.
It's my honor to present such information for the students.
Yeah, it was my honor to be host at this symposium for 2 days, and it was my honor to listen to this. So, dear colleagues, distinguished guests, and friends, and Dr. Ali Raza, we have officially reached the conclusion of the International Symposium on Biosafety and Laboratory Biosecurity Management. Over the past 2 days, we have shared incredible insights, learned from world class experts, and reinforced our shared commitment to strengthening safety, securing science, and protecting lives. I want to extend a final heartfelt thank you to our brilliant speakers, our dedicated organizers from VNIAS, USTB, and NUMS, our wonderful collaborators, and most importantly, to all of you in the audience for your time and active engagement. Thank you for allowing me to be your host. With this, I officially declare the symposium closed. I wish you all great success in your future research, and I hope to see you again soon. Thank you and goodbye.
Goodbye. Good night.